We will get started in just 1 minute. Thank you all so much for joining today's session. Good afternoon, everyone, and welcome to today's session, Moving Research and Discovery Forward. We are really excited for the first time ever to hold a live announcement on the results of our neuromuscular grant competition. My name is Hamera Osman. I am the VP of Research and Public Policy at Muscular Dystrophy Canada. Before I move forward too much into today's session, I'd like to make sure that you are all on the appropriate channel or the channel of your choice in terms of language. Today's session is offered in English and in French. So please go ahead and select the interpretation icon and select either English or French. As you all know, Muscular Dystrophy Canada's mission is to enhance the lives of those impacted with neuromuscular disorders by continually working to provide ongoing support while relentlessly searching for a cure through well-funded research. We know research remains a key priority for the neuromuscular community. So while we work towards providing supports in the here and now through a basket of programs and services such as funding for equipment and assistive technology, transportation, housing, employment, providing resources around education and system navigation, we are really pleased that we get to invest in tomorrow by funding research. This will truly help to improve health outcomes and influence positive change. I wanted to take a moment to really highlight your impact. Your support of MDC has allowed us in the last 5 years alone to invest $5.6 million on total research initiatives. We have been able to support research across 95 academic institutions. And that is about 115 projects that we've supported in the last 5 years, led by 115 lead researchers. Let's even drill in closer. Just in the last year alone, your support has allowed us to invest in seed grants. We were able to actually increase our number of investments in the last year by 116%. So compared to last year, we are able to support $800,000 in research this year. We are able to support an amazing network as a funder, as a partner, and really supporting a neuromuscular disease network, a pan-Canadian network, allows us to not only support particular research projects, clinical and translational projects, but allows us to support research and clinical capacity across the country. We are thrilled to announce that in the past year, together with NMD4C, we launched 2 new competitions. Clinical fellowship competition and a postdoctoral research competition. This is really investing in the next generation of clinicians and scientists. And so we were able to support 3 clinical fellows. These were individuals who trained in physiatry or neurology, who will now be receiving training in neuromuscular medicine, and 2 individuals who will be participating in neuromuscular research in the next year. We were able to answer your questions about your disorder, clinical trials, and we've had over 2,000 inquiries that have come through MDC's research hotline. So I'll take this moment to remind you all to please email research@muscle.ca with any questions you have. At MDC, we fund the best research, the high impact research. And we do this in a stepwise process, in a process in that we initially launch a call for clinical and translational projects. Researchers like the ones you'll hear from today write grants. And this year we received 24 grant applications. We then lean on a group of academic and community reviewers. This year, we engaged with 21 individuals who reviewed, rated those applications. The clinical grant competition was chaired by Dr. Hans Lochmuller, and the translational research grant competition discussion was chaired by Dr. Robin Parks. So I'd like to take a moment to thank both of them for their leadership on conducting the peer review discussions. And I wanted to mention, in terms of the community reviewers at MDC, the core and the strategy of everything we do is for the person impacted and their family members. So we were pleased to have individuals with lived experience also reviewing and rating the applications that came in. And that led us to, they helped us to narrow down those projects to 9 projects that we are able to fund as part of this competition. So I'd love to pass over the special announcement to our Director of Research and Evaluation, Dr. Daria Voitel. But Daria, don't you think this requires a drum roll of sorts? Over to you, Daria.
Thank you, Humira. It's my pleasure to congratulate the following researchers and their teams for being awarded research grants with MDC this year. Dr. Jody Warman-Chardon from Ottawa Hospital Research Institute, who'll be looking at quantitative mRNA imaging to assess the progression of OPMD. Dr. Carrie Schellenberg from the University of Saskatchewan, who'll be exploring the impact of spinal bulbar muscular atrophy on persons self-identifying as Indigenous. Dr. Miriam Osby from the Research Institute of McGill University Health Center, we'll be looking at motor network connectivity in spinal muscular atrophy. Dr. Michael Berger from the University of British Columbia will be looking at measuring balance in chronic inflammatory demyelinating polyneuropathy. And Dr. Reshma Amin from the Hospital of Sick Children, who will be looking at the transition of teenagers with SMA in a multidisciplinary adult program, and she'll be working with her colleagues in adult clinics around Toronto as well. And now from our translational grants, we are pleased to announce that our awardees in this stream are Dr. Bernard Jasmine from the University of Ottawa. He'll be looking at endurance training as a novel therapeutic for myotonic type 1. Dr. Craig Campbell from the London Health Sciences Centre in Western University will be looking at genome-wide DNA methylation profile in DMD. Dr. Christopher Perry from York University will be studying mitochondrial targeted therapies to improve DMD outcomes. And lastly, but not least, Dr. Thomas Durkin from McGill University, who will be building a screenable human 3D neuromuscular junction model for neuromuscular disorders. Over to you, Humira.
Thank you so much, Daria. Congratulations to all the awardees. Today, we've invited a few of our awardees to do a rapid research talk on their proposed projects. I've included an image or a vector here of a puzzle. Really, in terms of the research projects we'll hear about today, it's really one piece of the puzzle. Clinical and translational research together will lead us and move the needle forward and get us one step closer to identifying cures for neuromuscular disorders. Throughout the year, we'll also be showcasing the other awardees across our webinar series as well. But we'll go ahead and pass it over to Dr. Warman to start us off. Over to you.
Thank you so much for Humira and Daria. What a great introduction. I'm going to just share my screen. There we go. Everyone can see my screen now? Yeah? Okay. So I first wanted to thank you so much for being awarded the MDC Clinical Research Award. I was so excited and honored to receive it. And I realized there are a lot of outstanding applications that were submitted. The importance of this research is that we're looking for quantitative MRI. So this is an imaging biomarker to help assess progression in occulopharyngeal muscular dystrophy. So occulopharyngeal muscular dystrophy, or OPMD, is a muscular dystrophy that affects the eyes and the face and causes weakness of the eyelids and as well as difficulty swallowing, as well as a more limb girdle muscular dystrophy pattern in the arms and the legs. So difficulty swallowing, difficulty keeping eyes open, and then difficulty getting up and down stairs and getting out of chairs. But in Canada, OPMD is one of the most common muscular dystrophies. So elsewhere in the world, OPMD can be 1 in 100,000 or 200,000. But in parts of Canada, it's so common, it's 1 in 1,000. So this is a really important muscular dystrophy for the Canadian population. The big research gap, or one of the key research gaps in OPMD, is that there are several new therapies under development for many muscular dystrophies, including OPMD. But we really need better ways to assess progression, and more importantly, to assess people's possible response to these therapies under development. Right now, what we're looking at is how strong people are. So most people have had their doctors assess them at least once and check the strength in their arms and their feet, and that's very important for diagnosis. But it's just not close enough to assess for prognosis or for progression for many of these diseases, or if people are responding to therapies, especially for slowly progressive diseases. There's been a lot of developments in the muscle MRI. So a lot of people have had MRIs for their back or their neck, but MRIs can actually look at your muscles. So MRI is a really powerful tool to assess changes in the muscles and the progression and the distribution of which muscles are affected. There's no radiation with MRIs. It's not invasive. People aren't poked. It's readily accessible in many hospitals, and you can repeat it over time. And importantly, you can do the whole body at the same time. So in about 40 minutes, you can assess almost every muscle in the body in its entirety. And if you looked at the level of the thigh, so if this is at your thigh, you can see that each of the muscles have an individual pattern. And this would be at the level of the calf. So really, really important to see all these different muscles, and much better than our examination. In Ottawa, my colleagues, Dr. Sampaio, who's a radiologist, and Dr. Melkus, who's an imaging physicist, have looked at these new MRI techniques, and we can actually tell the progression of the muscle. So example, this is somebody's tongue with OPMD, and people often have trouble with swallowing. We can actually outline the tongue and tell how much percentage of fat or what that change of muscle, so how much the dystrophy has progressed. And with that information, we can actually assess all the muscles throughout the body at one point in time. And so as part of our study, the patients would be enrolled, and they would have an assessment with a neurologist. They would have some blood work done to look for other biomarkers that would correlate with the muscle MRI. They would have their MRI. So this typically takes between about 25 and 40 minutes. And then the doctors and the scientists are going through the MRI, and we can actually look at different levels. So the tongue, or the back muscles, or the leg muscles. And this is really critical to assess the progression of the disease. And so for the impact of this study, we're going to be able to look very carefully at how OPMD progresses with muscle MRI. And so we can look at the percentage of how the dystrophy changes. But really what we want to do is we want to get this population of patients with OPMD ready for when there are new therapies that are coming out. And so that this is a really critical clinical trial outcome measure. Importantly, this really positions us in Canada to have more studies for other neuromuscular diseases and other muscular dystrophies. So although the OPMD is the first group of patients, we'll be able to use this imaging technique for many patients with different neuromuscular diseases. And so in closing, I want to thank Muscular Dystrophy Canada very much for awarding the funding for this grant. We're really excited about how this is going to help change care and our ability to assess patients with OPMD and other muscular dystrophies, and how this can be used for other potential therapies and therapeutic trials in the future. Thank you so much.
Thank you so much, Dr. Warman. That was excellent. We will move on to Dr. Schellenberg now, who will be talking to us about the Kennedy's disease study.
Hi, everyone. Thank you so much for this opportunity to speak. I'm so grateful for the funding, and I'm so excited about this project upcoming, and to have the ability to share this with you at this launch is a wonderful opportunity. So I'd like to talk to you about photo voice in spinal bulbar muscular atrophy, in particular looking at it from an Indigenous perspective. So spinal bulbar muscular atrophy is a very rare disease. 1 to 2 per 100,000 is often the quoted prevalence. It's also known as Kennedy's disease, and it affects the lower motor neurons. So those nerves that exit from the brainstem into the face, and from the cervical and lumbosacral spine into the arms and legs. So it is inherited disease. It's X-linked recessive, which essentially means that the women carry the disease and the males become fully symptomatic. It's due to a CAG repeat in the androgen receptor gene, and essentially there's a abnormal polyglutamine, which is inserted into the androgen receptor gene, which causes it to be present in a mutant form in this disease. And so there are progressive neuromuscular signs that occur over time. So patients have difficulty with limb weakness and atrophy. And persons with this disease have difficulty with speech and swallow, and it can become more challenging over time, breathing difficulties. And one thing we're beginning to recognize is that this is a really multi-system disease, and it affects other organ systems such as increased risk of diabetes, fatty liver, and there are consequences of an abnormal androgen receptor, and this can cause infertility or reduced fertility and testicular atrophy and a number of other issues. So we recently published a study showing that the prevalence of SVMA in Indigenous communities in Saskatchewan was extraordinarily high. So I told you that the usual quoted prevalence is around 1 to 2 per 100,000. Here in our Indigenous population, it's more like 14 per 100,000. So that's 10 times the usual prevalence quote. Now, there are pockets of increased prevalence around the world, but the highest reported prevalence other than ours is in Finland, with a prevalence of 7 per 100,000. So ours is even double. And so, and I worry that this is underreported, because we know a lot of our patients who have cousins and uncles and brothers who either have symptoms and are not followed by medical care for a variety of reasons, or haven't been diagnosed yet. And so this is really a disease of important significance here in Western Canada. Now, Dr. Gerald Pfeffer's lab was the one who ran some of these genetic studies and showed that there was a founder effect, which essentially means that these patients had a common ancestor many years ago. And so with this, I introduce our research group, and without Dr. Alexandra King, there wouldn't be a project. She's the co-principal investigator on this project and is an Indigenous researcher herself, and is the Cameco Chair of Indigenous Health and Wellness at the University of Saskatchewan. Dr. Malcolm King is the scientific director of SCIPR, which is the Saskatchewan Centre for Patient-Oriented Research. And Dr. Gerald Pfeffer is a medical geneticist, a neurogeneticist in Calgary. And under the direction of the Doctors Kings, a community guiding circle was formed. And this is a group of patients or persons with this condition, as well as an elder Sharon Jinkerson Brass. And this guiding circle was formed to help understand the impact of the research and to direct the research. And so this community guiding circle really made it very clear that this was an important concern. They wanted to understand the impact of SVMA on their communities. They felt that there were many unmet needs that needed addressing, and that the first priority was to have a circle where they could share and talk about their experiences and hopefully gain a better understanding. And one of the research methodologies that really resonated with the community guiding circle was photo voice. And photo voice is a qualitative participatory research tool that is very patient-centered. It's increasingly used in healthcare settings. And it's been increasingly used and adapted for use in Indigenous contexts. And the reasons for this have been quite multifaceted. And one of the things that has been discussed is the visual storytelling approach and the equity-oriented storytelling approach that helps persons share their circumstances. And so with this methodology, participants take photos that illustrate their circumstances, so their lived experiences with SVMA. And this is followed up with individual interviews with patients and their families, as well as group interviews with other patients and caregivers with the researchers. Our clinic does have experience with photo voice, and I told you that it's increasingly used in healthcare settings. There are some studies done in neurologic disease. Ours was the first to show this research in ALS. And personally, I was overwhelmed with the stories of hope and resiliency that our patients showed. And I'd like to give you a flavor for the kinds of things that photo voice can bring out. So the first example, top left, it struck me like a crossroads in your life. You can either go to the left or you can go to the right. And I choose to go to the right. You've got to decide how you're going to deal with it, meaning ALS. And there's just not much point in being anything but positive. And then the second one of the major things ALS has stolen from me is time. It takes so long for me to do things now. Every action seems to be in slow motion. So the cracked watch really represents to me how my time is so fractured now. And this last example, this is where I eat. There's a pool noodle that helps to support the arms and the bent spoon so that I'm able to get food to my mouth, which is getting harder every day. You know, as a kid, you try to feed yourself. And as an adult, you don't want to have someone to feed you, but you do adjust. You can't be afraid to try new things, and you can't be afraid to invent. Our community guiding circle also shared many of their own struggles, the impact this has on themselves and their family, the impact that it has on their communities, the lack of support and lack of awareness. And they also discussed how, sadly, many of their symptoms have been misappropriated to lifestyle measures or lifestyle factors. And this can cause stigmatization and inappropriate care. And unfortunately, our CGC are not the only ones who feel this way. In fact, earlier this month, there was a Cree elder in northern Manitoba with Kennedy's disease, SVMA, who was asked to leave a furniture store because the staff thought he was inebriated. He wasn't. He had trouble speaking because of his neuromuscular disease. This just helps to, I think, really show the degree of advocacy and awareness required. So lastly, I want to say thank you to Muscular Dystrophy Canada for supporting this work. And I also want to thank our team, my co-PI, Alexandra King, as well as the full team that we've previously discussed that work with us. So with that, I will close. Thank you very much for your interest.
Thank you again, Dr. Schellenberg, and congratulations on this important research. I'll turn it over now to Dr. Craig Campbell to discuss his translational research project.
Well, thanks so much. It's wonderful to receive the award. I'd like to thank MDC and of course, all your generous donors who make these awards possible. Just trying to share my screen here. Why isn't that working?
Not a problem, Dr. Oh, there we go. I think I see it. It's just zoomed in a bit, but you are.
Let me see if I can do
There
this.
we go.
For some reason, it's wanting me just to do a portion of the screen, but let's see if we can right size this. Oops. Just have to pull this out a little bit. Does that capture it? Do you see the screen there?
That's great. Thank you so much. Go over to you.
Great. Yeah, thanks. And again, really appreciate sharing this session with the other grant recipients. So congratulations to them, and amazing work you're doing. So I'm going to talk on a translational research project that we're doing here in London around epigenetic signatures. This comes from a team of people, and in fact, even though I'm here to represent translational research, I think everyone will probably recognize I am not by nature a lab person. I really do mostly clinical research, but I've had the very great opportunity to work with Dr. Beckham Sadikovitz and his team in the molecular genetics lab here. And these folks are world leaders in epigenetic signatures and have really facilitated the work I'm going to show you that we have started with, and of course the work that the MDC grant will facilitate. And the other person I want to just do a shout out to is Leighton Schreier, who came into my lab as a BSc student and got quite interested in understanding some of the differences in the genotype and the phenotype that we can see in boys with Duchenne muscular dystrophy, and then we started to talk about different possibilities around that, and that led us to think about epigenetic signatures and capitalize on the work that Beckham and his team do here. Probably most will be aware that Duchenne muscular dystrophy is one of the more common muscular dystrophies we see, certainly on the pediatric side, and it's caused by a gene mutation in the DMD gene that causes a problem with this, very big protein here called dystrophin, and dystrophin acts as a almost like a scaffolding protein, basically that connects the inside of the muscle cell to the outside of the muscle cell. And when that is either absent or not working well, it results in progressive muscle weakness. And often the boys who have DMD are struggling with their walking and with their breathing. And I bring that up because I'm going to show a little bit of data related to that. So what is our study going to look at? Well, we've been working already in developing some pilot data, which I'm going to show you, which was the foundation for our application to the grant. And basically this work is really trying to understand if there is a genome-wide methylation pattern that we can associate with DMD, and then to understand if those epigenetic patterns in DMD are predictive of the disease progression and severity. So you may say, well, what are epigenetic, signatures, and what is epigenetics? In many diseases, we think of them as having an issue with a specific gene, many genetic diseases, and there's a mutation in a specific gene. But there is actually a whole layer of gene regulation that happens to turn genes on and off in specific ways that relate to factors other than just the sequence of nucleotides in the genome. And this extra layer of regulation of gene expression is called epigenetics. And we measure many epigenetic phenomena by looking at methylation of genes. And if genes have a higher or lower pattern of methylation around them, that actually can help us understand if those genes are turned on or off in a way that may impact a disease. And so that's what we are really interested in looking at in Duchenne muscular dystrophy, was beyond just the mutation in the dystrophin protein, are there patterns of genes that are turned on and off in a different way that would help us understand whether we can assign a signature to that, and whether the pattern of that high or low methylation of those genes help us understand the something about DMD that we didn't know before. So what we did is we started out by looking at a small number of boys, and our project is going to look at a larger number of boys, and looking at over 800,000 different methylation probes in the body, in our genome. And we found ones that were actually unique to DMD situation, that were either hypomethylated, which you see on this volcano plot here, many hypomethylated areas in the genome, and some hypermethylated regions. And that was the first indication that we may be able to take some of those regions and create a specific epigenetic signature, a profile of hyper and hypomethylated genes that would be specific to DMD. It also unlocked the idea that maybe we could look at many of these specific areas in the genome to see if they would be genetic modifiers of the disease. And I'm not going to talk too much about that today, but we've identified many genes that we might think would be involved in the muscle pathways that we're going to look more fully at. And in fact, with our small number of kids, we were able to identify a distinct epigenetic signature. And here in the graph, you can see that small number of boys in red who have DMD, and they're and this is based on about 118 probes that we found very distinct patterns of methylation compared to a group of control samples. But we need to do more children with DMD to really understand if this pattern is consistent and robust. We need to examine outliers from this and see if there's patterns that might in kids with DMD that don't fit here. So far, all we've done is blood tissue, and we'd like to obviously look at muscle because we know that's where some of the significant problems in DMD lie. And then we want to try and bracket some of our findings with kids with more mild forms of muscle disease like Becker's muscular dystrophy, look better at an age range and whether these patterns change over age, and then looking at how steroids impact this epigenetic signature, because we know steroids can cause changes in methylation patterns in some of these regions. And then the other big pattern or hint that we saw was, could these methylation patterns actually end up being associated with changes in the disease severity? And again, in this small number of patients, what we tended to see was that as people's weakness progressed and their ambulation status changed from walking to being in a wheelchair, we saw the epigenetic signature look more distinct, which is what you see in this graph. And then we saw the same pattern with respiratory status, that as you became more dependent on respiratory support, the epigenetic patterns seem more distinct. And so we want to try and understand this, because this could be something we could use to understand disease progression. And so ultimately, what we are hoping to show with our studies is, can epigenetic signatures be used as part of the diagnostic process? There's many areas of the world where we can't get gene sequencing, at least not rapidly or cheaply, and epigenetic signatures may help in understanding that. Again, as I mentioned, do hypo or hypermethylated sections of the genome help us to then understand the pathophysiology? And we've started to try to pull this apart a little bit more in our lab. Can we use epigenetic signatures to understand disease progression and prognosis? That's an intriguing thing, which would add another new biomarker. And we've actually also already started to talk with different industry partners to look at using epigenetic signatures pre and post treatment in some of the novel therapeutic trials we're doing to look at whether it could be a marker of treatment responsiveness. But there are a number of knowledge gaps that this grant is going to help us to really flesh out the steroid contribution to the epigenetic signature. We want to make sure the steroids are not a confounder. The biomarker issue of disease severity responsiveness to treatment, looking at different genotype-phenotype relationships. So there are unique scenarios where two children have the same mutation in the dystrophin gene, but have differences in how their strength is or how they're doing. And we want to see if these epigenetic signatures could help us understand that. And then again, more at understanding whether there are hints in the epigenetic signatures that can help us understand modifier genes. So thank you very much. Again, much gratitude to MDC for the grant and to the donors who have made that possible. Thanks for letting me be a part of this today.
Thank you very much, Dr. Campbell. We really appreciate your leadership on this important project. We'll hand it over now to Dr. Reshma Amin, who will talk to us about the transition project for SMA.
Hi, good afternoon, everybody, or good morning or good evening, depending on where you are across the country. I just want to echo the speakers before me in that myself and our team are incredibly grateful for the funding support of Muscular Dystrophy Canada for our project, and we really appreciate the invitation to speak to you all today. So I'm going to be speaking about our project, which is the transition of teenagers with spinal muscular atrophy to a multidisciplinary adult program, the Teen SMA Study.
Dr. Amin, I hate to interrupt, but before we move forward, do you mind just swapping? Great, so we can see it on full screen. Beautiful. Go ahead.
Apologies, such a rookie mistake this far into the pandemic. All right, so just a bit of background. So spinal muscular atrophy is an autosomal recessive neuromuscular disorder with a prevalence of about 1 to 2 per 100,000 people. And it's a disorder that affects the motor neurons of the anterior horn. It's estimated that approximately 1/3 of the SMA population are adults, and this proportion is only going to continue to grow with the disease-modifying therapies and improved therapy overall for this patient population. With regards to transition from pediatric to adult care in general, we know this is a time that's been associated previously in literature with poor clinical outcomes. So during this transitional phase, patients are at risk of having increased healthcare dropout, so being lost to clinical follow-up, having poor treatment adherence with therapy, having sort of a decline in their overall wellness with increased illness severity, and then also having an increase in hospitalization. At present, unfortunately, SMA transition or multidisciplinary adult service programs do not exist uniformly across the country or really largely at all. And as the care of this growing population becomes more complex, multidisciplinary care is strongly recommended to really try to parallel what we do on the pediatric side. So, of course, this is a huge clinical gap for our patients across the country. So in response to the current unmet needs, SMA transition and adult multidisciplinary service programs are being initiated and implemented at multiple centers in the Greater Toronto area. So at Sick Kids, Holland Bloorview, Sunnybrook Health Sciences, and also West Park. And of course, this implementation offers an opportunity for simultaneous evaluation. And we want to learn from this and really understand the patient and family experience. And we want to learn from patients who are newly transitioning to adult care and their families, but we also want to learn from patients who have already transitioned to adult care to hear about their experiences thus far. So for our study objectives and our goals for the study, we wanted to, as a primary objective, explore the experience and readiness for transition for teenagers with SMA and their families. As a secondary objective, we wanted to explore the lived experiences of adults with SMA and their family caregivers that have already experienced transition. And then as an exploratory objective to help sort of inform future transition studies and evaluations, we wanted to explore the impact of transition on healthcare use and costs, quality of life, mental health, caregiver burden, as well as clinical outcomes. So with regards to our study methods, so we've planned to do an 18-month prospective study. So we would follow patients who are newly transitioning from pediatric to adult care for a period of 18 months. It's a mixed method study, which means we're going to be using a mix of sort of quantitative methods like survey measures, also with interview style data collection as well. We're going to be enrolling patients from all across those 4 centers in Toronto. And in terms of study participants, we're going to be having 2 groups of patients. So we'll have the first group where it's adults with spinal muscular atrophy that have already transitioned to adult care. And then the second group will be groups of teenagers with their primary caregiver, and we'll be following that group forward as they transition through adult to adult care. On this slide here, I've highlighted what will be the infrastructure for the clinical transition program. We'll go through this first and I'll take you through the research plan. So it'll be a series of 4 visits. For visit 1, this is sort of a visit in the 17th year. The pediatric team is present. During visit 2, which is the last pediatric visit, the pediatric team is present and the adult team will also be present either virtually or in person. There's also going to be a 3rd visit, which will be a joint transitional visit where both the adult and pediatric teams will be present simultaneously. And then visit 4 will be after the transition, and that will be an adult clinic visit with just the adult team. We've tried to be opportunistic and build in the research framework to parallel what's happening clinically for these patients. So cohort 1 are the patients that are adults and have already transitioned. So those patients would be interviewed after an adult clinic visit and after they're consented. But for cohort 2, this is the cohort of patients that we're following forward. Patients would be enrolled when they're still a pediatric patient and then followed forward over time. And there'd be multiple types of data collection in terms of just collecting information about their clinical status. Also information in the form of qualitative interviews, so semi-structured interviews to hear about their experience with the healthcare system at different points in time. A questionnaire that asks about their transition readiness, questionnaires that look at their perception of coordination of care, as well as questionnaires from the perspective of the caregiver, looking at caregiver burden, and then also questionnaires about quality of life. So what's the so what here? So we're hoping, our aim is that our findings are going to be informing best practice for patient-oriented models of care for spinal muscular atrophy, but also for other neuromuscular disease populations in Ontario with hopeful generalizability across the country and also internationally. And also through identifying and learning from patients and their families about their experiences, we hope to be able to identify barriers, facilitators, and indicators of perceived readiness for transition that will then be able to sort of integrate back in an iterative way to then continuously improve transition moving forward. And of course, the ultimate goal is to improve clinical care over time and to improve the experience of transition as well as health-related quality of life of these patients and families. I just want to highlight our large team of people that have contributed to this work and that are going to be contributing on an ongoing basis. So our team, just to highlight it, is a mix of patients, clinicians, trainees, and researchers, and now, of course, Muscular Dystrophy Canada as well. And of course, I'd just like to echo the generous support of Muscular Dystrophy Canada, and thank you all for your time today.
Wonderful. Thank you so much, Dr. Amin. At this time, I'd like to invite our researchers back on to start their videos, and we'll start a fireside chat about the impact of the funding and also open this up for any questions you might have around those research projects as well. I do see one question coming in. One question is, talking about what about congenital muscular dystrophy. And I saw a similar comment also in the chat about a different disorder. So today we heard about projects related to OPMD, Kennedy disease, Duchenne muscular dystrophy, and spinal muscular atrophy. And I think at MDC, we're in a privileged position where we represent a broad set of neuromuscular disorders, and we're able to invest in research that is disorder specific, but just as you said, Dr. Amin, that has an ability to transfer, and the results can relate to other disorders. But I'll pass it over to you, Dr. Warman, about how these studies can help to inform clinical care for other neuromuscular disorders.
I think, all of the studies that were funded are focused on one disease, but I think the importance is that they can be used for many different diseases. So for example, for my MRI study, and I also see a lot of patients with congenital muscular dystrophy that are adults or recently diagnosed. And so the imaging study that we use for OPMD, it will be the same type of sequences and the same type of methodology. So we can use that for many different muscular dystrophies, including congenital muscular dystrophies. And I'll let my colleagues also speak, but I think that all of the different research studies that were funded can be applicable to many different diseases just by the methodology that's been funded. I don't know if Dr. Campbell wants to, in particular, mention about the GWAS, about how that can be attributed and.
Yeah, I would echo that, Joy. I mean, certainly this has opened up a little bit of a door for us in trying to understand epigenetic signatures in other neuromuscular conditions. We know it has some roles in myotonic dystrophy. That's been looked at a little bit, but this is really an area where I think hopefully we'll be able to be instructive and informative for other conditions. So thanks for stretching us into thinking about that.
Wonderful. The next question that has come through, and I acknowledge that one of your patient research partners is in our audience today, Dr. Amin, but the question is for you. You've included patient research partners in your research project. Why is that important? And if you can also describe what a patient research partner is.
Sure, absolutely. I think that more and more as clinicians, we're coming to base the focus, and for many of us, we're clinical researchers, really the direction that we take and the focus that we take in our clinical research program really reflect what we're hearing in the clinic from our patients. And so for me, myself, I think when I'm in clinic, I hear what are the struggles for my patients. They help me understand what the gaps, because I see one perspective of the pie as a clinician, but I have no idea what it's like to be the one to navigate the healthcare system. So I think there's always a role for patient involvement in research. And I think more and more across the country, that's really been a focus. That's one of the great things about MD Canada and the ongoing partnership with clinicians and nonprofit organizations and access to patients that we have through them. And then I think in terms of, I think having patient involvement in clinical research can take the form of several different kinds of things. So I've been involved in projects where actually it was my patient's idea to actually focus on something and do that type of research project. I've been involved in other projects where patients and families can comment on the protocol and become partners, both sort of in the phases of development and then follow up. So I think it really just depends, and there's sort of no right or wrong fit. And I think more and more there's even formalized organizations at many of our institutions that have patient organizations for research-led input directed by patients and families.
Absolutely. And once again, that definitely aligns with our mission here at MDC and our strategy. And I think I can take this opportunity to share that this year we will be rolling out or launching training on how to become a patient research partner, thanks to the work that's happening with the Neuromuscular Disease Network. And that work package is led by Dr. Kathy Selby. So that is really exciting. The next question is for you, Dr. Warman. We wanted to take this time to congratulate you as you recently recently were awarded a CIHR, a Canadian Institutes for Health Research grant on burden of inherited neuromuscular diseases. And that is a grant for a larger amount. But what we wanted to ask you is, what is the benefit or value of these seed grants, or receiving funding for seed grants, as you've also received larger funding as well from government agencies?
So what a great question. So the smaller grants, or the information, these are still very, very generous grants. But what we can do with these grants that are typically done in one center is that we can develop the infrastructure and some of the pilot data or the information to convince granting agencies that might commit, $500,000 or $1 million towards neuromuscular disease research. So when you're donating or supporting an organization such as Muscular Dystrophy Canada, your research can increase tenfold, because these grants can be used as pilot data for larger grants that can be done across the country and with international partners. So, we really appreciate the funding that we get for these grants, for these really exciting projects that's going to help care and improve research for patients with neuromuscular disease. And these will lead to much larger grants and the data that we really need to apply for these larger grants to improve the research in neuromuscular disease.
Wonderful. Thank you so much. A question for you, Dr. Schellenberg, and it is also, I'll open it up to the others as well. A few individuals in the comment section are asking about how to participate in your research. How do you often recruit for a study like the one you described today? And how can people hear about these research opportunities?
Oh, I always love to hear that people want to participate in our projects. Those are always wonderful questions. So we have a number of projects ongoing in spinal bulbar muscular atrophy. And so and you don't necessarily need to be part of our clinic in order to participate in many of these cases. We need to connect in some other ways. I think the easiest way would probably be to call our clinic, 306-655-7742. But we have a number of ways that we can integrate participants from a number of different provinces. We've already connected with other provinces in both West and East in order to increase our recruitment pools and to allow more provinces to participate.
Fantastic. Dr. Campbell, I know you're here today as a clinician, but also leading a translational research project. Can you speak to why it's important to invest in translational or early stage research as well?
Yeah, thanks for that, Humira. I mean, these kind of projects that take us from, you hear the term, from bench to bedside, are really projects that take what is happening in the basic science lab and figure out and fill in the the gaps that are going to need to move it to something that can be used in a clinical way that can make an impact day to day. Unfortunately, things don't just jump from the lab right into actual practice. We often need to put some foundations around those things, whether they're drugs or new diagnostic tests like what I've described today. But often there is a set of of questions and knowledge that needs to be laid down about how something that looks so promising in the lab will actually make an impact in people and in the clinic. And that can mean sort of carefully testing if it's a drug. It means carefully testing the safety and how the drug distributes in the body. If it's a diagnostic test like what I've described today, it can be looking at a small set of people and looking at different disease characteristics and really understanding is the test performing the way it should before it's rolled out to a larger study, and then hopefully into clinical practice where it actually helps change how helps us to be better around a certain aspect of muscular dystrophy. So that's how I think about translational research. Maybe the other little thing I'll say, is having a clinical scientist, a clinical person connected to a basic science person really allows us to do the kind of conversation that helps us do the best science, right? Because a lot of times our basic science colleagues have a really in-depth knowledge of some of the mechanisms and things that as clinicians we don't always appreciate, and helps us understand what things we have to think about, and vice versa. So really that conversation and having that translational research conversation helps move our whole community forward.
Absolutely. Thank you so much, Dr. Campbell. And just to add to your remarks, I would say at MDC, when we're thinking about translational research, we're thinking definitely from bench to bedside, but also to community, to thinking about how we can use some of these research findings to influence public policy as well, and in terms of access to therapies and treatment. So this is fantastic. Thank you all so much. I'm going to now turn it over to our CEO, Stacey Lintern, for some closing remarks. Over to you, Stacey.
Thank you, Homira, and hello everyone, and thank you for joining us today. And thank you and congratulations to all of our presenters. The Canadian neuromuscular community is so fortunate to have dedicated, passionate, and brilliant researchers conducting research in the neuromuscular field. Research is powerful. For the first time, neuromuscular treatments and therapies are available. We know that with advancements and breakthroughs, as we've heard today, in one neuromuscular disorder, there's incredible opportunities to transfer this knowledge and advance other breakthroughs for other neuromuscular disorders. Research is an enabler. Research is critical for influencing positive changes with policy so that the right resources are available for the neuromuscular community at the right time. And research is hope. Muscular Dystrophy Canada will continue to work relentlessly in partnership with the research community to find cures. Certainly, Muscular Dystrophy Canada is so grateful for the support of the neuromuscular community. Your support enables Muscular Dystrophy Canada to move the needle forward for neuromuscular research. Thank you to the thousands of donors, fundraising volunteers, SHADs, R&R, and firefighters. You make it possible for us to continue to invest in research that mobilizes treatments, therapies, standards of care, public policy, and ultimately cures. Thank you to our Medical Scientific Advisory Committee for your guidance and the many peer reviewers who volunteered their time to review and select the research projects. On June 11th, we will be hosting our Walk for MD virtual event. We are extremely excited as Dr. Wharton is the Walk for MD Honorary Chair. We are so grateful to have such a prestigious researcher partner with us. Dr. Wharton is joined by a team of brilliant, dedicated, and committed research champions who will be raising funds for Muscular Dystrophy Canada so that we can continue to invest in research. The Walk for MD fundraising goal is to raise $1.3 million. Not an easy task, but as you know, necessary. Please register for the Walk for MD by going to walkformd.ca or dystromarch.ca or email walk@muscle.ca. We have staff who are available to support you through every step of the way. If you'd like to sponsor Dr. Wharton and his incredible team, please email walk@muscle.ca or you can contact me directly. As you know, at Muscular Dystrophy Canada, we do not receive government funds for core mission activities, so every donation counts. If you would like to donate in a different way, please reach out to me. It's our priority to make it easy, and we would be grateful for you to join us in making a difference for the Canadian neuromuscular community. On behalf of the thousands of Canadians affected by neuromuscular disorders, thank you from the bottom of our hearts for your support in advancing neuromuscular research and making a difference. We are grateful. Take care.
Thank you so much, Stacey. On that note, I do see Dr. Wharton in the audience today, and I thought it would be timely to again invite everyone to the Walk for MD and to end on a quote that he has shared with us. Canada has some of the best researchers in the world, many of them at the forefront of new technology development, and the prospects for treatments are more promising now than ever. Every dollar raised for Walk for MD takes us closer to the day when many of these disorders succumb to innovative treatments, not even imaginable in 1985 at the dawn of gene discovery. We thank you again for your support and for attending today's session. Congratulations to all the awardees. If we weren't able to get to your question, we'll be able to answer these offline. And again, welcome any questions and you can direct them to research@muscle.ca. Truly, thank you on behalf of Muscular Dystrophy Canada. Thank you all.